My name is Joseph Munyaneza. I'm with U.S. Department of Agriculture Research Service. I'm stationed at Wapato in Washington. I have been working on zebra chip for a very long time. We have been dealing with disease epidemiology, but specifically I've been working on potato psyllid, which vectors the disease. So one of the things we have been doing was to try to understand how these insects transmit the zebra chip bacterium, Liberibacter, and how these result into zebra chip development. So up in the Pacific Northwest where I'm located, we have 3 genetic variants of potato psyllid, which are very common there. We refer them to haplotype. So the 3 haplotype we have in the Northwest are Northwestern, Central, and Western. So what we have been finding out is if you go in Idaho, in Washington, and Oregon, you usually have different haplotype of psyllids being there in a potato field. The problem is, even though we know that these psyllids are genetically different, we're not sure if they have the same biological traits. I mean, can they transmit Liberibacter to potatoes the same way? So we did a study to see, to effectively check on that, to see whether one of the haplotype is more efficient in transmitting Liberibacter. And so what we found out was that the 3 haplotype, which are common in the Pacific Northwest, they have the same feeding behavior and are, the efficiency, the transmission efficiency of the bacterium is the same. Basically, there is no difference in the transmission of the bacterium and causing zebra chip by the 3 haplotype, which means for growers, doesn't matter which haplotype you have in your potato field, whether it's Western, Central, or Northwestern, you just have to go ahead and treat them. So they seem to be transmitting they do transmit the same way. Another interesting thing we found out when we were doing this study, we found out that these insects, they are so effective, they can basically, when they land on a potato plant, it takes them about 2 hours. After 2 hours, they are able to transmit the bacterium. And once they start infecting, so one thing I forgot to mention is the bacterium is phloem limited. So the bacterium has to be deposited into the phloem to cause zebra chip. And so the psyllids, when they land on a plant, they start probing and they don't reach the phloem until 2 to 3 hours. But once they get into the phloem, it takes them about less than 5 minutes to transmit the bacterium and cause zebra chip, which is very significant because it takes a very short period of time to transmit the bacterium, and you have to, if you're going to control these, you have to come up with something which is going to kill these insects quickly, very rapidly, or prevent them to feed.